Hello, I'm Yu Liu, professor at Shanghai University of Sport. I am honored to have this opportunity to share my work in the area of neuromechanics. In today's talk, I will focus on two major areas of my research. First, I will introduce some of the ongoing studies that we have been conducting over the past 4 years that specifically look at how neurostimulation-based techniques can be used in the context of sport performance enhancement. Second, I will talk about some of the field work involving applications of neurostimulation that we have been doing in collaboration with the Chinese National Sports Commission and the National Winter Sports Teams as they prepare for the upcoming Winter Olympics in early 2022. I would like to begin my talk by acknowledging my research team members for their dedication, commitment, and diligent efforts in conducting and promoting neuromechanical research and SUS. Neuropriming commonly includes invasive and non-invasive approaches that involves the application of electrical stimulation to drive or alter neural function within a circuit in the brain. In my talk, I focus on non-invasive based approaches such as transcranial direct current stimulation, alternating current stimulation, transcranial magnetic stimulation, and a new way, namely temporal interference stimulation techniques. Neuropriming techniques have received considerable attention in recent years, with applications ranging from clinical treatment for many of neuropsychiatric and neurological disorders to training to enhance and accelerate military soldiers' abilities in navigation, attention, and motor skills to training to specifically boost athletic performance in sports. So the neural mechanisms of how non-invasive brain stimulation modulates brain activity and enhance outcomes are still not well understood. It is generally believed that non-invasive brain stimulation modulates neuronal activity and subsequently alter neuronal excitability of brain regions involved with a behavior of interest. Other mechanisms are influence decreases in gamma aminobutyric acid concentrations and increase brain-derived neurotropic factors that facilitate synaptic plasticity and network connectivity in the brain. In 2017, Grossman proposed a temporal interference stimulation. It can provide non-invasive stimulation of deep brain regions of mouse. Over the past 3 years, we have been engaging in a systematic research efforts that focus specifically on 1. Protocol development and the initial validation of TDCS, TACS, and TI-based techniques for understanding their ergogenetic effects on conditioning and cognition-related human performance. 2. Further protocol validation and mechanistic studies via animal models. 3. Brain imaging tests in human subjects that specifically examine biomarkers of key characteristics and mechanisms related to neurostimulation interventions. And 4, conducting neurostimulation studies, compare different non-invasive brain stimulation techniques on athletic performance. Here, I would like to emphasize that our research approach takes a fully integrated neuromechanical mechanics paradigms to understand ergogenetic effects of neural stimulation on athletic performance and the mechanisms underlying non-invasive brain stimulation, which represent a significant step forward in biomechanics research aimed at understanding human performance in sports. In recent years, more and more attention has been paid to the research and application of TDCS in sports science. Also, there are different opinions on the conclusions and applications of the TDCS for improving sport performance. Many studies have been shown that it can improve muscle power, extend the time to exhaustion during exercise, and promote motor skills learning and acquisition. However, how to choose the stimulation parameters and the position, especially the scientific combination of the training program, still need to be paid great attention in the future. This slide shows our study results of a TACS on working memory and biomechanical control of dart-throw task. This is the findings. You can see the working memory is significantly improved after a TACS protocol test. The TI stimulation of motor cortex induced neurophysiological responses. The excitability of motor cortex is significantly improved after a TI stimulation. And present fMRI technology can accurately locate the brain function activation area. Using resting fMRI can also study the functional connectivity between different brain regions. Combining fMRI with diffusion tensor imaging, magnetotopographic transcranial magnetic stimulation, and other technologies can obtain more information about the brain function activity. This slide shows our preliminary results of neuroimaging study of TI stimulation. The TI stimulation on the left primary motor cortex, figure 1, significantly enhanced the functional connectivity, figure 2, between the motor network and the left prefrontal cortex. This pattern was observed in 2 independent experiments using different simulation protocols. Now, I'm going to talk about the application of neurobiomechanical techniques to improve performance in Chinese Winter Olympic athletes, funded by the China Ministry of Science and Technology Winter Olympics projects. We are providing scientific support and service for national teams, including, for example, ski jumping team, snowboard U-track skier team, freestyle skiing area team, alpine skiing team, figure skating team, and so on. For example, in ski jumping team, we use full high-speed cameras to take high-speed video of the ski jumpers' takeoff phase and perform 3-dimensional motion capture by using AI algorithms, analyze and compare the kinematics of the athlete's takeoff movement, feedback the monitoring data to the coach team, and provide good support. The coaches analyze and guide the athlete's technical training. This slide displays the interface of our ski jumping 3-dimensional model system. At the same time, there are statistical data of takeoff and flight process on the right side. The video shows the digital athletes during ski jumping, driven by our 3D motion capture analysis data. and we can provide coaches and athletes the biomechanical feedback data of takeoff, like distance for takeoff, knee angle, and the trunk angle, and the attack angle, and the takeoff phase. This is a fast feedback report given by our ski jumping intelligence analysis system on a certain training days. The fast feedback information to coaches and athletes. This slide shows the speed, time, and the distance history of one ski jumper in the whole flight process, monitored by using UWB and IMU wearable sensors. You can see that the maximal velocity and the maximal acceleration during takeoff, flight, and landing. The maximal ground reaction force of the athletes can also be estimated during takeoff based on biomechanical model. In terms of ground reaction force, it is about 2.3 times body weight, which is correlated to vertical velocity of takeoff, a very significant indicator of performance according to the coach's guidance. This is an ongoing study of our neurobiomechanical enhancement techniques. We will focus on, in the future study, first, personalized stimulation protocol, and closed loop smart regulation, and third, the physical cognitive improvement in order to get realizing the Olympic motors on higher, faster, and stronger. This picture shows the neurobiomechanical enhancement technologies and SUS. Thank you for listening.